Hi, my name is Lauren Hoffner. I'm going to be going over the journal article, Aptamers Generated from Cell Selects from Molecular Medicine, a Chemical Biology Approach. So in this research, they wanted to they generated cell-based aptamers to understand the molecular basis behind cancer cells, because molecular medicine focuses on the molecular basis behind diseases, and in order to diagnose and treat the diseases. So what are aptamers and cell selects? Aptamers are single-stranded DNA and RNA oligonucleotides that fold into secondary and tertiary structures. These secondary tertiary structures react to target cells with high binding affinity and with the specificity of the way antibodies react with these target cells. So cell selects is the method that is used to make these aptamers. And what happens is there's a single-stranded DNA library you use. You incubate the single-stranded DNA with target cells. You wash and remove unbound sequences, extract the bound sequences, react them with negative cells in order to remove sequences that wouldn't bind with the target cells. You collect the unbound sequences from this part, and you amplify the sequences using PCR in order to characterize the aptamers. And this process is has many advantages over using natural made antibodies, because aptamers are easy to synthesize. You can chemically modify them depending on what therapeutic method you're trying to do, and they are not toxic. So for their research, in order to make cell-based aptamers from cancer cells to observe the binding affinity, they use CCRF-CEM cells as the positive control, or the target cells, and the Ramos cells as the negative control. So the CCRF-CEM cells come from acute lymphoblastic leukemia, while the Ramos cells comes from Burkitt's lymphoma. So what they did was, after they basically collected the DNA products they got from the cell select cycle and labeled them with fluorescence labels, and incubated the DNA products with CCRF-CEM cells and Ramos cells. And it was observed that the fluorescence intensity increased for the CCRF-CEM cells, while the fluorescence intensity did not change for the Ramos cells. So as the rounds kept going, the binding affinity got more and more enriched and specific, and the Ramos cells didn't change. So the whole experiment was very successful. And based off this technique, they were able to make cell-based aptamers from other types of cancer cells. And as I mentioned earlier, these aptamers can be chemically modified, and that makes them very good candidates for cancer diagnosis and therapy, because as you can see here, this is an original aptamer, and then this is the truncated version of it, and the binding affinity actually went up for the truncated version of it. So like maybe you can bind it to specific to unknown biological markers and to other drugs. So the most interesting experiment I thought they did was one that involved targeted chemotherapy, in which they used this aptamer, SGCHC. They covalently linked it to doxorubicin, a anti-cancer drug, and basically made this conjugate, and it possessed properties of high binding affinity and were able to penetrate the endosome of these cancer cells here. And what they did was, they incubated the conjugates with the cancer cells, and then A, B, and C represents like the time frame. So A is at 30 minutes, B is at an hour, and C is at 2 hours. So as you can see, as time passed, the SGC8C docs were able to penetrate the endosome of the cancer cells and target these cells specifically. So this is really important, because if you could just target specific cells with chemotherapy, the chemotherapy won't kill cells that aren't cancerous, and the patient may experience less toxic side effects that come with chemotherapy. So they basically did many more experiments, and some of them included, they were able to form two methods to detect cancer early, and they were able to find unknown biological markers. So this research is really important for molecular medicine, and I thought it'd be cool to share with you. So thank you so much.